Have you ever stopped to think about how something as seemingly simple as a bacterium can be so, well, complex? Today, we're going deep, deep down to the microscopic level to take a tour of the incredible molecular factory that builds the very proteins these tiny organisms need to survive.
It really all boils down to this one question. A cell has its genetic blueprint, its DNA, right? But how in the world does it turn that simple, elegant code into thousands of different complex molecular machines, the proteins, that do literally all the work? It's an absolutely mind-boggling feat of engineering. So, to get our heads around this, let's imagine we're taking a tour of this molecular factory. This is where the real magic happens, It's where that abstract genetic code gets translated into the physical stuff that makes life, well, life.
Okay, so the factory itself, it's called the ribosome, and this thing is a beast. It's a massive, super complex machine made of both protein and RNA, and its one and only job is to build all the other proteins the cell needs. It is the absolute heart of the whole operation.
Now, any good factory needs a set of instructions, a blueprint. In our case, that's messenger RNA, or mRNA for short. Just think of it like this. It's a temporary copy of one page from the cell's master DNA blueprint, and it's carried right out onto the factory floor.
And finally, you've got to have your raw materials and a way to get them to the assembly line, right? Well, that's the job of transfer RNA, or tRNA. Each one of these little tRNA molecules is like a specialized delivery truck, and it's responsible for bringing one specific type of building block, an amino acid, to the ribosome.
All right, but before our factory can even think about starting up the assembly line, it has to make sure all those delivery trucks are loaded with the right cargo. This step is so fundamental, we could call it the zero step of the whole process.
So how does this actually work? Well, there's this incredible enzyme that basically works like a super-specific cargo loader. It finds and grabs a particular amino acid. And then, using a bit of energy from a molecule called ATP, it hooks that amino acid onto the correct tRNA delivery truck. Only when a tRNA is charged up like this is it actually ready for the assembly line. It's a crucial quality control step that makes sure the factory always gets the right parts. Okay, our delivery trucks are loaded up and ready to go. Now it's time to fire up the main factory. This is what we call the initiation phase, where all the big machinery comes together at the perfect spot to kick off production.
So with this long mRNA blueprint, how does the ribosome know where to even begin? Well, in bacteria, it looks for a special little landmark called the Shine-Dalgarno sequence. You can think of it like a neon sign that says "Start Assembly Here." It positions the ribosome perfectly, just upstream of the official start codon, the three-letter genetic word that kicks off the whole protein recipe.
And the way it all comes together is just fascinating. First, the smaller piece of the ribosome grabs onto the mRNA blueprint, with a little help from some special proteins. Then, the very first tRNA, the one carrying the starter amino acid, is brought right into place. It's only when everything is lined up just right that the bigger part of the ribosome clamps down, and boom, you have a complete, functional factory. It's a whole series of checkpoints to guarantee a perfect start every time.
And now we're at the main event, elongation. This is the assembly line running at full tilt. It's this super efficient, repetitive cycle that just keeps adding amino acids to the growing protein chain. And you won't believe this. This little machine can add up to 20 amino acids to that chain every single second. It's almost unbelievably fast. So the cycle kicks off when a new, charged tRNA truck pulls into an open bay in the factory. We call this the A site. You can think of it as the arrival bay. It's where all the new parts are delivered, and they're even escorted by a little helper protein just to make sure they get to the right place.
Once that new tRNA is locked in, the ribosome does what it does best. It acts as a catalyst, and it forges a super strong chemical link. That's the peptide bond between the new amino acid and the chain of amino acids it's already built. That growing chain, that's our brand new protein in the making.
So with that new link made, the entire assembly line kind of lurches forward by exactly one position. This movement is called translocation. It shifts the tRNAs over, boots out the now empty tRNA truck, and, crucially, opens up that A site for the next delivery. And just like that, the cycle is ready to go all over again. But wait, how does the ribosome keep things so accurate while moving at that incredible speed? It's all about quality control. It checks the delivery slip, making sure the three-letter code on the tRNA, its anticodon, is a perfect match for the code on the mRNA blueprint. If it's a perfect match, the ribosome physically changes its shape, locking that tRNA into place. It's this amazing physical check that prevents the wrong parts from ever getting added. And that forward movement, translocation, it's not just a simple slide. Oh no, it's a really complex coordinated dance. There's a special protein called elongation factor G that comes in, uses a burst of energy, and acts like a molecular ratchet, physically pushing the mRNA and tRNAs through the factory. It's this powerful, precise mechanical action that keeps the whole assembly line chugging forward.
Of course, the assembly line can't just run forever. It's got to stop somewhere. It eventually reaches the end of the blueprint. And this final phase, termination, is all about stopping production cleanly and, really importantly, getting the factory ready for the next job.
So how does the factory know when the protein's done? Well, the mRNA blueprint has these special three-letter words called stop codons. And they don't call for an amino acid. Nope. Instead, they're like a big red stop sign. And they're recognized by specialized proteins called, you guessed it, release factors.
When one of these release factors slides into the A site, it's like a supervisor hitting the big stop and release buttons at the exact same time. It makes the ribosome use a water molecule to slice the finished protein free from that last tRNA. The completed protein floats away, the job is done, and the entire factory disassembles, its parts ready to be recycled for the very next job.
Okay, wow. We've just been on a whirlwind tour of a very, very tiny and very busy factory. But why does any of this matter? Why is it so important for us to understand this incredibly intricate process inside bacteria?
Well, it's because this factory is the absolute undisputed engine of all bacterial life. Without it, bacteria can't build a single thing they need to survive or to replicate. And that right there makes it a perfect target. You see, many of our most powerful antibiotics work by specifically jamming the gears of the bacterial ribosome, shutting down their protein factories without harming our own. So the better we understand this machine, the better we can get at designing new drugs to fight disease.
There's a quote from the research that just sums it all up perfectly. It describes the ribosome as a dynamic molecular machine. And that's exactly what it is. It's not some static lump. It's constantly moving, changing its shape, checking its own work. It is a truly stunning piece of natural nanotechnology.
So the next time you think about a single bacterium, don't just picture a simple little blob. Picture a bustling, microscopic city, packed with countless molecular factories, just like the one we saw today, all working non-stop to turn a simple code into life itself. Which really makes you wonder, what other incredible things are going on in there?